Year 11, this is a short video to go through some of the ideas surrounding mitosis and meiosis. We'll also touch briefly on the use of stem cells and what they are. Okay, so first of all, big questions. We know that new cells are produced by old cells dividing, but why are these new cells needed? Well, basically, they're needed for two different processes. One, they're needed for growth and repair, and the second one they're needed for is production of the sex cells. We also call the sex cells gametes. Okay, so new cells are needed for two main reasons. So if they're needed for two main reasons, there must be two types of cell division, mitosis and meiosis. Now during the tutorial we'll basically break down and compare these two types of cell division. So if we start with meiosis, now meiosis is a type of cell division that produces gametes, or the sex cells. In the case of humans, that would be the eggs and the sperm. Okay, so if we start with the parent cell, now meiosis takes place in the sex organs. Okay, and the parent cell is going to contain a diploid number of chromosomes. So in this case, it's going to be 46 single chromosomes or 23 pairs. Now the first thing that has to happen is the chromosomes have to make identical copies of themselves. They then line up in similar chromosomes. We call them homologous pairs. Now sometimes sections of DNA can get swapped to increase the variation. And then the pairs of chromosomes divide, so they move to opposite sides of the cell or poles of the cell. So this is the first division. Now in meiosis there is then a second division where the chromosomes line up, and again they are pulled to either side of the cell, so they're separated. So we end up with 4 daughter cells, okay, and each of the daughter cells are genetically different. Now what you'll also notice, because the cell has divided twice, that we started with 23 pairs of chromosomes. We then doubled the amount of genetic material, so we could say that we've got 92 single chromosomes. It divides once and then divides again, which leaves us with 23 chromosomes in each. Okay, so each of the 4 daughter cells have 23 chromosomes. So if the sex organ was the ovary, then the 4 daughter cells would be eggs. If the sex organ was the testes, then the 4 daughter cells would be sperm. Okay, now the reason that meiosis has to halve the number is so that during fertilisation the diploid number, the full number, is maintained. So there's 23 pairs of chromosomes or 46 chromosomes in total. Just think a little bit about sexual reproduction. So we know that the gametes are formed through a type of cell division called meiosis. We know that in humans the sex cells are eggs and sperm. We know that the eggs and sperm contain a haploid number of chromosomes, so half the number, so 23 chromosomes, and during fertilisation the nuclei of the sperm and the egg fuse together, and what they form is a fertilised egg, also known as the zygote. Now in terms of development into a human being, the next thing that happens is that zygote that now contains 46 chromosomes, or 23 from the mother, 23 from the father, now has to grow and divide to produce an embryo. Okay, to do that, the cells are going to divide. Again, because it's for growth and repair, we are using a different type of cell division called mitosis. Mitosis, we have a slightly different process, okay? So the first thing that happens is we start with the parent cell, okay? And again, what's going to happen is all the genetic material is going to double, okay? The chromosomes then line up at the equator, okay? And then spindle fibres attach to the chromosomes and they pull them to opposite poles of the cell. The daughter cells, you can see this time we've only made 2. Okay, and they are genetically identical to the parent cell. So if the parent cell had 46 chromosomes or 23 pairs, the daughter cells also had 23 pairs or 46 chromosomes. So just a reminder, it's used for growth and repairment of cells. The genetic material is copied. The cell divides once to form 2 genetically identical body cells. And this is how the embryo is created. Now, within that embryo, there'll be a part of the embryo that basically contains what we call embryonic stem cells. Now, embryonic stem cells are unspecialised, and what that means is that they can become any type of cell in the human body. So they could become muscle cells, blood cells, nerve cells, skin cells, hair cells. And when they differentiate as the embryo develops, they become specialised. Okay, and that basically means that they are adapted for a particular function. Okay, so muscle cells, for example, are adapted. They've got lots of mitochondria, so they can contract and relax. A sperm cell has a tail. That's what we mean by differentiated. Okay, so unspecialised cells are part of the embryo. We call these stem cells or embryonic stem cells. As the embryo divides and as the embryo develops, some of these stem cells will go on to specialise to become different types of cell. Now, what scientists want to do is they want to use stem cells in treatment. So they want to take stem cells, they want to harvest them from a variety of different places. So embryos, umbilical cord, and also you can get it from adults in their bone marrow. And they want to harvest these stem cells so they can treat particular diseases. So because stem cells are unspecialised, they can be made by scientists to differentiate into cells that might treat these diseases. So for example, we could take out stem cells from embryos, we could turn them into nerve cells, and then those nerve cells could help treat somebody with Parkinson's disease or paralysis. Now, we can also take stem cells from umbilical cords or bone marrow. Now, these are what we call adult stem cells. Now, they're not as good as embryonic stem cells because they can only differentiate into a few types of cells. So, for example, stem cells that are removed from the bone marrow can only develop into different types of blood cells, so white blood cells, red blood cells, etc. Now, there are social and ethical issues, particularly concerning human or the removal or harvesting of human embryonic stem cells. And it's this idea that basically you are killing or destroying a potential life to remove those stem cells. Okay? Whereas to take stem cells from the bone marrow, it's a simple operation, and obviously the umbilical cord goes to waste, it's not used. So they are the simple ideas about mitosis, meiosis and stem cells.